Hello, overcomers, and welcome to this episode of Connect Over Coffee. I am Rinsi, the founder of Overcome, and today we are joined by our very special guest and very special friend, Dr. Amir Jazaeri. So Dr. Jazaeri is the Vice Chair for Clinical Research and the Director of the Gynecologic Cancer Immunotherapy Program at MD Anderson Cancer Center. Regarded as one of the top global experts in this space, Dr. Jazaeri has been involved in some groundbreaking work in ovarian cancer that is here to share with us today. We have a lot to chat with him about all the promising advances being made in the world of immunotherapy, not just immunotherapy, but just ovarian cancer in general. And so join us for the next 45 minutes to an hour as we chat with Dr. Jazaeri on this insightful conversation. And please share this, as I always say, please share this video and this information far and wide with anyone that may benefit from all the pearls of wisdom he's going to share with us today. And so I have my beverage ready. Hope you have yours, and let's connect over coffee with Dr. Jazaeri. So with that, a huge welcome back to you, Dr. Jazaeri, to this episode of Connect Over Coffee. Our overcomers always love to hear from you. And I do.
Thank you so much for the opportunity. I love to connect with Overcome and the overcomers, and I send out my greetings to anybody who is joining us virtually.
Thank you. So, again, I always have a lot of questions, and you all always say I have sub questions to every question, which I do. But in starting with the basics, right? There are many therapies out in the market, which is promising, which is hormonal therapy, targeted therapy, immunotherapy. So just before we get into the depths of the immunotherapy and such, tell us just briefly, what are the differences between these three kinds of therapies, the hormonal, the targeted, and the immunotherapy, and how are they used, and which overcomers would benefit the most from each kind of therapy, and at what points in time, when are they used in the treatment cycle? So see, I have questions and sub-questions. So.
Yeah, no, I love it. I love it. And thank you for that really good question. We could probably easily spend a whole hour on that one question alone, but I promise I won't do that. So as many of your audience are aware, different cancers are treated with different types of treatment. And that is because those particular treatments have found to work best for those cancer types. And so diving into ovarian cancer, for example, we know that initial treatment with a platinum chemotherapy drug and another drug called Taxol often works very well for ovarian cancer, especially in the beginning. And that's why our initial treatments use those two drugs. Sometimes even when the cancer comes back and it's been a long time since the patient has received those two drugs, we sometimes go back to those two drugs or combination of a platinum drug and another chemo drug. So definitely chemotherapy plays a long role in terms of long-term outcomes for patients with advanced ovarian cancer. Surgery, of course, also plays an important role, but it's mostly surgery and chemotherapy, but with an emphasis on chemotherapy for longer-term outcomes. And you notice I didn't mention radiation. So sometimes patients with ovarian cancer say, well, could radiation play a role? Because of the spread pattern of ovarian cancer, the fact that it has access to the entire abdominal cavity and even in the chest cavity, and radiation being really good for cancer that's limited to one region or one area in the body, we don't use radiation very frequently for ovarian cancer, and we rely more heavily on chemotherapy. So that's an example of a disease where chemotherapy plays a large role. And I should mention, when I say ovarian cancer, I'm really referring to the more common high-grade ovarian cancers. In contrast, patients that are diagnosed with low-grade ovarian cancer often see hormonal therapy as part of their treatment. They also may receive surgery and chemotherapy, but the biology and the clinical behavior of low-grade ovarian cancers are such that make hormonal therapies particularly useful. And hormonal therapies are basically drugs that attack the estrogen pathway or other hormonal pathways. And again, oftentimes they might be in oral form, in pill form, but sometimes they can be injectables or other drugs. So I would say within ovarian cancer, there's definitely a role for hormonal therapies, but largely limited to those patients that have low-grade ovarian cancer. And last but not least, of course, immunotherapy has been a big game changer for many cancer types. I would say we still haven't quite figured out what combination of immunotherapy drugs works best in ovarian cancer. That doesn't mean that we've given up, and certainly that remains a large focus of our cancer, is finding out the right types of immunotherapy and immunotherapy combinations for patients with ovarian cancer. So right now, there's nothing that's FDA approved, but there is a combination that's listed in the NCCN guidelines. So for your audience, the way we can prescribe medications for cancer patients that are then often approved by their insurance is that these must be FDA-approved indications, or kind of an alternative route, if you would, is these are drugs where there's enough evidence to be listed in national guidelines, such as the NCCN guidelines, but maybe there isn't the critical information required at the level of FDA approval. And so the combination of three drugs, Avastin, Pembro, or another name for it is Keytruda, and oral cyclophosphamide or oral Cytoxan, which is a chemo oral drug. This combination can be used in patients with recurrent ovarian cancer, although it's not an FDA-approved indication. And so I hope that I've given you a little bit of a flavor of different kinds of treatments that can be used in patients with ovarian cancer. But I think the overriding principle is we use these tools when they're most applicable, and some might be more applicable in the beginning when, for example, patients are chemosensitive. Chemotherapy certainly makes sense. Later, during stages where the cancer has become more chemo-resistant, then we might use a combination such as the immunotherapy combination that I mentioned. And then for certain cancer types, such as low-grade ovarian cancer, hormonal treatments might be beneficial.
Thank you. And so you talked about the 3-drug combination, and this is a great segue into my next question. So because I was going to ask you, about the therapies that does involve the combination of 3 or more drugs, are they generally effective in terms of balancing the outcome and the toxicity? And so talk to us about that a little bit. And can you share a few examples of such treatments currently being used in and out of the clinical trial realm?
Yeah, I think there was a time in ovarian cancer where we considered, well, maybe we can try different chemos. More chemos might leave less chance for ovarian cancer cells to become resistant to any one of the drugs. So, there was a trial that took place about 20 years ago where instead of 2 chemo drugs, patients were treated with combinations of 3 or more drugs. And unfortunately, that trial was a negative trial, meaning the outcomes were the same, regardless of whether patients got the 2 best drugs, the carboplatin and the Taxol, or if a so-called second-line drugs such as Doxil or gemcitabine was added to that backbone. And so we know that solving the ovarian cancer problem is not as simple as, oh, let's just add more drugs, because if that worked, then we might say, well, we might accept the additional toxicity, but unfortunately, it seems that it's not that simple. But I think that sort of principle of why are we adding drugs? Are we getting more benefit? And if so, is that benefit at a risk of increased side effects? Should be a discussion that patients feel very comfortable having with their oncologist, because there's always a flip side to the coin. We might add. I think a common example of adding a third drug is patients that might have recurrent ovarian cancer and are being treated with a platinum drug and another drug, and then bevacizumab or Avastin is added to that combination. And some of that is based on trials that showed that when you do that, patients live longer. But I think, in practice, we sometimes extend those strict indications. We may have a patient that's already been on bevacizumab, and should we then continue that bevacizumab even though the cancer progressed on that drug, just because a previous trial showed benefit? I think these are questions where we don't have scientific sort of answers to, but I think we can certainly apply common sense and logic, and also I think it's important to remember that not everybody responds to drugs in terms of side effects the same way, and some patients may tolerate a certain drug, and somebody else might have a much harder time tolerating that same drug. So I think the guiding principle is more is not always better, and that the side effects and the downsides should always be discussed with patients when we recommend a combination. And that recommendation is usually based on evidence. It's not because I think adding these two drugs might be useful for my patient. These are based on trials that have shown benefit. But again, even those trials may or may not apply, because if you were already on one of those drugs, then maybe the trial that shows 3 drugs is better than 2 drugs may not really apply in that specific patient.
Yeah, so physician discretion as well as the unique disease state of the patient both together will determine what the approach.
Disease state and treatment history of the patient. Yeah, absolutely.
Makes sense. So last time when you were on, we talked about the role of, you were working on the PPP2RIA hotspot mutation. So yeah. I know several advances have been made. So can you tell us about the advances that has happened in this space of your work, and what our overcomers should know about this particular indicator and its progress to impact the disease?
Sure. So let me just bring up to speed some of your overcomers who might be hearing about this for the first time. So some number of years ago, we identified patients that were as part of one of our clinical trials where we were testing an immunotherapy. Those patients with clear cell ovarian cancer, about 10% of them have a mutation in a gene called PPP2R1A. And these patients tend to do better than those patients that didn't have the mutation. The problem was 90% of patients don't have this mutation, only 10% do. So the strength of our conclusions were not super strong, but over time we've gathered more information. We think that there's definitely a story there. And we asked the question of these mutations, inactivate this pathway. What if there was a drug that did the same thing? Then that drug might be beneficial for the 90% of patients that don't have the mutation. And wouldn't that be worth testing? And I'm glad to report to everyone that we now have such a trial open. Again, we kept it in clear cell ovarian cancer because our original observation of benefit was in those patients. So, I think some of the immunologic sort of reasons for why this drug that inhibits the same pathway might work in a broader population definitely are there. But we wanted to show in the same patient population, can we have a positive impact on those clear cell patients who have recurrent platinum-resistant disease, don't have many good standard options, but might be able to participate and get two immunotherapy drugs. One of them is a commonly used immunotherapy drug called Pembro or Keytruda, but we're combining this drug with LB100, which is the drug that attacks the same PP2R1A pathway, but as a drug, so you don't have to have that mutation in your tumor. So this trial is open at MD Anderson, and we hope to open it soon at Northwestern because one of the people who worked on this trial with me is now a faculty at Northwestern, and so we hope to kind of spread the word, especially for those patients that have recurrent clear cell ovarian cancer, that this might be an option for them, for those that have developed chemo-resistant disease. And, so if that trial is positive, then we can ask questions like, can this work for other patients that have other subtypes of ovarian cancer, not just clear cell? But that's, of course, down the line.
So very promising, but I'll take you back to this because I want to dig deeper a little bit because a lot to take in from all the information you just shared. So I think you mentioned in the beginning that this, first of all, this is only for the clear cell community. And then of the clear cell, I think you mentioned that only 10% of the clear cell ovarian cancers have this mutation.
Mm-hmm.
So the trial that you have open right now, I think you also mentioned that it is also for the people who do not carry this mutation. So
in fact, it's only for people who don't have this mutation because we feel like, well, we know the mutation has a positive impact. To really look at the impact of the drug, we had to take the 90% of patients with clear cell that don't have the mutation. So technically, if you have the mutation, you're not eligible. But again, those patients that have the mutation, their oncologists can try the 3-drug immunotherapy combination that we spoke about just a few minutes ago, the combination of bevacizumab or Avastin with Keytruda or pembrolizumab and the oral chemo drug Cytoxan. That's something that is in the standard, I say standard because it's not FDA approved, but it's listed in NCCN guidelines for use. So patients that have the mutation, that could be a potential option for them. For those patients that don't have the mutation, we're doing the trial to see if this investigational combination might provide benefit.
So that's great. So 90% of the patients then under clear cell community could potentially sign up for this trial. And so where are you in the the recruitment stage, and how many patients are you recruiting? Because one of the questions we always get asked is that there are often not too many open positions. So how many patients?
Yeah, that's a good question. As you know, clear cell is a relatively rare subtype of ovarian cancer, so it's always more challenging to recruit patients with clear cell. Also, we're looking at patients that haven't already had immunotherapy, so that narrows down our potential population further. Yeah, I can say that we've recruited about 4 or 5 patients. The total accrual is about 21, 22 patients. So there's still plenty of room for those that are interested in participating. We're in the process of developing a video. One of the things that we're always interested in is how do you take this medical information and make it more accessible to patients, and how do how many times have you heard from overcomers that they hear some news about their disease, maybe not responding to the treatment, and who doesn't immediately get on the computer to Google what's the newest and latest and greatest for their type of cancer. We're hoping to make these videos that have to be IRB approved, so we need to make sure that the content in the video is consistent with the information and the protocol, and doesn't overpromise or anything like that. So there's definitely sort of it's not just putting together a video and putting it out there, but we're hoping to have that IRB approved soon, so that we can share it with organizations such as Overcome, but also post it in other sites where patients with ovarian cancer may do searches or reach information, and hopefully get the information to the patients that need it the most.
That's wonderful. And so for this patient community, which you just talked about, if they're interested right now to be, to inquire about the trials that are open, then they should reach out to MD Anderson or Northwestern, because these are the two locations that are currently open, right?
Yeah, and Northwestern is getting ready to open. I would say right now, MD Anderson's the only site that's open. We anticipate Northwestern opening in the next 3 months.
Okay, wonderful. Thank you. So then, talking about promise, next question brings me to the drug factory implants. I know you are also very heavily involved in this promising project, and I traveled together 2 months ago for this presentation. So in terms of the current state of the trial that started with so much promise a couple of years ago, right? Where is this? From what I understand or read, it seems like the trial is not currently recruiting at this point in time. So tell us a little bit about the challenges and the roadblocks, as well as the successes and learnings that you've had with this trial, and where are we right now with this particular drug factory implant, and then how is the future evolving?
Yeah, so that's a great question. As you know, this is really exciting. So these cells, or so-called drug factories, can produce biologically active anti-cancer treatments that can, be implanted inside the abdominal cavity. That's relevant for ovarian cancer because that's where the cancer tends to grow and settle. What we saw in the phase 1 study was that these can be safely administered, but that the duration of activity, the duration where they make, we were interested in an immunologic drug called IL-2 or interleukin-2. This drug has a lot of side effects when you give it intravenously, but can be more safely administered intra-abdominally, and of course these factories were able to really produce them in biologically meaningful concentrations without many side effects, and so we are in the process of reviewing all of the clinical information from that project, which kind of showed feasibility, but also really highlighted that we need to come up with adjustments to make this last a lot longer, right? It's hard to treat ovarian cancer in a matter of days or weeks. You need a way to extend that benefit. And so we're working with Omid Veza from Rice University and some of the other bioengineers to see what are ways that we can extend the benefit, both in that cell factory model, but also Omid was the recipient of a large grant from ARPA-H, which is a new government agency that's really interested in funding innovative, high risk, high reward types of projects. And so it sounds a little bit like science fiction, but the next iteration is to maybe have a device where these cells are encapsulated. They have an oxygen supply so that they can live a lot longer inside the body, maybe for months, and then still secrete anti-cancer drugs. The device can also has electronic communications, so it might be able to talk to a cell phone or other smart device via Wi-Fi communication, and that can provide the study team information about what's going on in the tumor microenvironment, and basically try to give us more information about cancer. Right now, information we get from cancer are every few months when we do CT scans. And obviously, the cancer is continuously evolving, growing, responding, not responding, resistant cells building. So there's a lot going on between those gaps of, every 3 months where we do a scan. And right now we're blind to all of that. And so the idea is, can we somehow monitor what's happening at the tumor level more closely so that when the cancer goes around the treatment, we can also respond at the same speed. And so that's kind of what's down the line. We're hoping to have the first iteration of this device ready for human testing in hopefully January or February of 2026. So not this coming year, but in about a year and a half. Of course, there are very brilliant engineers that are working on making the device, making sure it's going to be safe in people, and a bunch of work has to be done before we have the privilege of testing it in people. But we're all very excited about this because we feel that this could really shift the balance of information and reaction to changes in the cancer environment.
Thank you for that brilliant answer. So just so that, because we do receive inquiries and questions from our overcomers about this particular trial, because there was a lot of hope and promise in it. So right now, from what I understand, the phase 1 is sort of completed with this trial, and so this trial is on pause. So at this point, or at this time, there is no additional recruitment happening. So the next phase would be in early 2026. So
yeah, there may be some in between. Basically, the trial that's currently no longer open, the initial phase 1 trial, that those cell factories are now have been acquired by another company, and so I think it remains to be seen if further development might happen in that setting as kind of a bridge between the trial that's already completed and the device that we hope is going to be start testing in about a year and a half. So there is a possibility that there might be sort of another investigational trial, either continuation of the original trial or a separate trial that might take place in between these two time points, but that remains to be seen.
Okay. And so we will just stay in touch with you, and we'll relay back the information to our overcomers should that trial open up, because we do receive those questions consistently. So thank you for that. So then for the phase 2 that you said that potentially will start in 2026, who would be the optimal candidates, or have you are you not there in terms of designing that yet, in terms of which overcomers? Or
yeah, I think again, we always have to make sure that we're not introducing investigational treatments which are largely an unknown, right? All clinical trials, all investigational treatments should be taken as promising yet unproven. So So I think just sometimes there's confusions. When I speak to patients, we want to make sure we don't ascribe unknowns. While promising, we don't want to overvalue them, right? Because the truth is we don't know. So these trials are usually introduced into patient populations that have recurrent and platinum-resistant disease. Why? Because we know those patients have fewer options, fewer useful standard options. And so it makes sense to offer those patients something that might be promising but unproven, whereas somebody who's newly diagnosed with ovarian cancer, we would not want to put something unproven and promising ahead of standard chemotherapy. So my guess is, while the study is still, obviously under development, I think that the patient population will probably be patients with recurrent platinum-resistant disease, and obviously there'll be probably a number of other eligibility criterion, but that remains to be determined.
Thank you. So before I switch gears, while we are on the immunotherapy topic of immunotherapy, what other latest advances are happening in this field that our overcomers should know of, and what is happening in Jazaeri lab, which is exciting and promising?
Yeah, so I think one of the sort of types of immunotherapy that maybe you hear or your overcomers might be less familiar with or hear less about is drugs like Keytruda or pembrolizumab are in the class that are called immune checkpoint inhibitors. These drugs work on immune cells inside of our body. And sometimes the analogy of taking the brakes off is used to say these drugs kind of decrease the brakes that can be applied to our immune cells by the cancer cells, and therefore help our immune cells better recognize, attack, and kill the cancer cells. And so these are drugs that are infusions, usually given intravenously once every few weeks. In that sense, they're closer to, their administration is similar to chemotherapy, even though their mechanism of action and side effects are very different. So there's a whole different class of immunotherapy where we actually are dealing with immune cells. These immune cells can be taken out of the body, expanded into large numbers. They can even be genetically modified, engineered, so they can recognize a specific target on the cancer cells, and then placed back into the body with the hopes that they're going to go and find and attack and kill the cancer cells. And these are called cellular therapies, or adoptive cell therapy is another term that's used. These cells are often the patient's own T cells, which are a class of immune drugs. But one of the sort of limitations is for the T cells, the patients have to basically undergo a procedure called pheresis, where it's kind of like giving blood, but it lasts 4 hours, and they take out some of the white blood cells out of the body. That becomes the starting material. Then these cells are engineered and expanded and returned to the patient. So that's one type of cellular therapy. Patients may have heard the term CAR T cells. That's kind of one type of cellular therapy where it works like that. Something that we've been working with one of our colleagues at MD Anderson, Dr. Katie Rezvani's lab, has been pioneering the use of a different kind of immune cell, the so-called natural killer, or NK cells. These are very powerful immune cells in our body, and for reasons that I won't get into, it's easier to give NK cells that might be from somebody else. So you don't need to use your own NK cells, but NK cells, in fact, that are used by Dr. Rezvani's group are from cord blood. So Of course, many of your overcomers are, familiar with, umbilical cord blood, and that this can be banked and stored. As it turns out, the umbilical cord blood has a large number of NK cells because it's very protective for the infant and the newborn. And so they use these NK cells to produce a cellular therapy that is off the shelf, meaning you don't need a lot of preparation. Patients that require it, and just like T cells, these NK cells can be engineered to recognize specific targets on the surface of cancer cells. And so, sort of one of our exciting trials is in collaboration with Katie Rezvani's group, where we're using NK cells that recognize a protein on the surface of ovarian cancers that's called TROP2, TROP2. And this is a cancer target that's already used in breast cancer. There are drugs in breast cancer that target the same target. And we and others have shown that this target is relevant to multiple other tumor types, ovarian, pancreatic cancer, for example. And the other cool thing is that we are administering them through a port directly into the abdominal cavity. I've already mentioned sort of ovarian cancer residing in the abdominal cavity several times during this conversation, but it's also relevant to many other cancer types. Pancreatic cancer, for example, can spread into the abdominal cavities, gastric, colorectal cancers. So it really has relevance to other cancer types too. And we're interested in this sort of different way of administering the immune cells directly into the cancer environment, which in this case is the abdominal cavity. So that's kind of something that I'm excited about. We have a trial where we're testing it in pancreatic cancer, but also in patients with platinum-resistant ovarian cancer. Again, starting with platinum-resistant disease because we want to show benefit potentially there. It's too soon to know, but that's something that's exciting. And it's administered through a port. Many of your overcomers might have portacaths that is underneath the skin and the chest. The other end of that port, of course, goes into the big blood vessels that run under the collarbone. In this case, the radiologist places a port that's placed on the lower part of the chest, and the other end of that port goes into the abdominal cavity. And that port then can allow us to introduce investigational therapies into the abdominal cavity, but it can also allow us to put in sterile saline and aspirate, and so get cells out of the abdominal cavity, kind of like doing a biopsy with each treatment. So participants in this trial undergo this port placement. We collect fluid and cells before we start the treatment, and then periodically every few weeks on the treatment to see what's happening during the treatment. So whether it works or not, we can understand what is happening with the immune environment. If it's working, how can we make it work better? If it's not working, then what can we do to counteract whatever is preventing it from working? So this is something we're really excited about, and again, kind of doing it in collaboration with experts at MD Anderson.
That sounds really promising and very exciting, as you said. So how many patients do you have on this trial right now?
Right now we've done the first 2 dose levels. These types of cellular therapies, you have to, of course, prove safety first. The study is designed for 3 dose levels. So we've completed dose levels 1 and 2 without any sort of concerns, and we're now starting the dose level 3. And again, we're sort of seeing this as an initial approach, but already working on second generation of these engineered NK cells that target TROP2. There are ways to make them less sensitive to immunosuppressive proteins that might be present in the abdominal cavity and in the tumor. And so, again, we kind of see this as an initial but iterative approach where, each study is going to sort of hopefully provide incremental benefit that gets better and better over time. But of course, you gotta prove, the proof is in the pudding, as they say.
Fascinating, though. Thank you for sharing. And
Yeah.
so now I just wanted to ask you about sounds like from what I've read about your work that you are kind of refocusing on the second look laparoscopy, right?
Yeah.
And so tell us about the evolution of this and why did you decide to re-explore?
Yeah. Yeah, that's thank you for mentioning that. That's something that's kind of near and dear to my heart. So you know, I think many of your audience recognize that one of the things that makes ovarian cancer so difficult to cure is not the initial treatment. The initial treatment, actually, with surgery and chemotherapy, we can make many patients enter remission. Remission means when we no longer have any evidence of cancer. So CA-125 tumor marker becomes normal. CT scan doesn't show any disease. And yet, the problem is that after that successful initial treatment, the cancer for many of those patients returns. And when we say returns, it's kind of a semantic thing. It doesn't really return. It's that the cancer never left the body. And so, there used to be a time many decades ago where the duration of how long should that first chemotherapy last was determined by a second surgery. That second surgery followed the initial tumor debulking, so it was called a second look operation. That second surgery was also done through another big vertical incision. Multiple biopsies were done, and if any of those biopsies showed residual disease, patients then continued on chemotherapy. The problem was, this is in the 1970s and 80s, the number of drugs that were useful for ovarian cancer back then and number of drugs that were available even as investigational treatments were quite limited. Maybe you could count them on one hand. And so as you might expect, the surgery was much more sensitive than CA-125 or CT scan in determining whether there was this residual disease. And even patients that had residual disease detected and got more chemotherapy didn't unfortunately translate into longer overall survival. And so this was kind of relegated to investigational uses and kind of fell out of standard practice for many decades. But what it meant was basically we were accepting that, well, unfortunately, if we detect that there's residual disease there, well, that's too bad because we can't do anything about it. And of course, what's changed now is we have so many other investigational treatments, so many that we don't know how to do clinical trials with. There's just too many things and permutations and combinations. So we can now do these operations through laparoscopic surgery or minimally invasive surgery, where they're now outpatient procedures. So patients come in and go the same day. And yet, even in 2024, what we've noticed from our sort of initial experience at MD Anderson is still surgery is much more sensitive than CT or CA-125. So it definitely, the detection of disease is much, much greater. And for those patients that have disease, as you might expect, they have a much worse prognosis compared to those patients that don't have disease. And we are asking the question, should we be intervening at this point when we know these patients have a poor prognosis and we know this is the lowest number of cancer cells in the body, right? At this point on, the cancer is just going to slowly grow and grow until it becomes clinically detectable. And really, kind of the conventional wisdom in the field has been, well, it does no good to treat ovarian cancer if it's at that low level because it's not going to make any difference. And I think some of us refuse to sort of accept that it's not going to make any difference without showing it. Let's see if we can intervene at that time point. Let's see if we can use some of the treatments, novel treatments that are now being used in the recurrent platinum resistant setting. What if we use those earlier? Can we eradicate this so-called minimal residual disease, or can we at least make patients cancer-free for a lot longer period of time? And so these are some of the kind of unconventional wisdom types of thinking. And we were fortunate enough to get funding from an organization called Breakthrough Cancer, where they're trying to bring together 5 large institutions: MD Anderson, Sloan Kettering, Johns Hopkins, MIT, and Dana-Farber, to collaborate. And this was one of the projects that they picked, is not just detecting minimal residual disease, but understanding what is the nature, what is the biology of this maybe 0.1% of ovarian cancer cells that are left behind, maybe even less. Because, of course, this also is where chemo-resistant disease begins, right? These are cancer cells that have lasted 6 or 7 treatments with chemo. So naturally, all the chemosensitive cells are going to die off, and what's going to be left behind is going to be this chemo-resistant population. And so the idea is to do these surgeries, both to more accurately communicate with patients. I think a lot of it is, I was feeling almost dishonest with patients saying, well, hopefully you'll be in the 10% of patients that might be cured. But, the reality is that's not likely for a large portion of patients. And we call them in remission, but we actually know that there is a way to actually prove that the cancer is still there. And if we can offer investigational treatments at that point that don't have too many side effects, then, hopefully we would be intervening sooner in a meaningful way. But of course, all of that needs to be proven. So right now it's research, and we're trying to also understand what is it about those 0.1% of cells that are left behind, so we can develop more directed treatments specifically against that minimal residual disease phase of ovarian cancer.
It is so hopeful that you all are working in in this space, because one of the questions I generally routinely have asked many times on our Connect Over Coffee series is exactly what you're saying, is once the treatment is over and the patient is in remission, knowing that for majority of ovarian cancers, the cancer returns or are never left, as you said, how can we tackle that while and not wait for the cancer to come back and do a scan or a CT like every 3 months to figure out how the cancer has progressed? And so I've always asked that is there nothing that we can do in between to not kind of prevent the cancer from coming back? And the answer I repeatedly kind of receive is like, but even if we knew what the state of the cancer was, what are we going to do with that information is what I hear. But it sounds like from your perspective, you all, through the Breakthrough Cancer Initiative, you all are actually trying to find an answer to that what to do. And yeah.
We're trying to attack it, and it's kind of the perfect storm in a good way, right? So we're interested in re-looking at second look surgery and biopsying areas of minimal residual disease. These biopsies will allow us to learn the biology of minimal residual disease. There is now many assays that can be done on small biopsy material that hasn't always been the case. You needed large amounts of tissue to do sophisticated analyses. now on a single microscope slide, you can look at expression of all the genes in the body, and you can see how different immune genes are interacting with cancer cells. And, so you can learn a lot of insights from just a single section. So that's another part of the perfect storm. Yet another part of the perfect storm is, of course, there's a limited number of second look and third look and fourth look. We can't keep looking surgically, but there's now technology in terms of liquid biopsies and tumor DNA that can be detected in the bloodstream that might be able to help us. The central problem of doing a trial in a patient that has no disease on a CAT scan is, then how are you going to know if it's going to work, right? Which is a perfectly legitimate question. And typically the answer is, well, we're going to follow those patients until recurrence. But those trials take a long time. And what we're interested in is use some of these blood-based MRD assays, these circulating tumor DNA assays, because for those patients that are positive, that can be a starting point. These assays, I should add, don't just say positive or negative, they also give you an amount. So if we can substitute the amount of cancer on a CAT scan to the amount of cancer by ctDNA, a blood test, then we can do interventions and expect that the promising interventions are going to result in decrease or disappearance of tumor DNA in the bloodstream. And so that might be really the thing that we need as a proof of principle that, look, we should be investing more in these patients that have minimal residual disease. And we don't need to do large phase 3 trials where placebo and active agent are followed until progression. These are very expensive, large, multi-institutional, multinational trials. Maybe with smaller, more nimble trials, we can show that if you add drug X, Y, or Z to patients that have detectable MRD by ctDNA, you can see reliably that level of tumor DNA decrease. And maybe that is something that's worth putting the additional resources and investing in to do a formal, phase 2 or phase 3 study. And I think that's what's going to be needed, because the good news is we have so many treatments these days and an ever expanding list of potential treatments. But there's not enough time, space, money to investigate it all as fast as we want, because we want difference for our patients' lives as soon as possible. And so we think that this MRD space also can play a large role in accelerating discovery in ovarian cancer.
I love this, and it truly seems as a breakthrough for ovarian cancer. So congratulations on this initiative, and that you're working with so many, all the top hospitals in the nation. And so we'll be definitely following the Breakthrough Cancer Initiative and
Absolutely.
finding out what's happening and bring you back for more progress updates. So I know we are almost reaching the top of the hour, but I know that you are. So we talked about your work on the clear cell ovarian cancers. Is there anything else that you would like to share in this space? Because we do have a very strong and active community in clear cell that wants more answers.
Yeah, no, I think, I shared with you sort of the exciting stuff that we have. We talked about the NK CAR program, the clear cell stuff, the MRD stuff. Of course, there are numerous other investigational treatments. Some are targeted therapies and depend on certain biomarkers being present on individual patients' cancers. Some might have biomarkers, some don't. But as always, I think you always give this advice to your patient, to your participants and your community that don't be afraid to reach out for second opinions. Don't be afraid to reach out for clinical trials. And I think that's how we together can really improve outcomes for patients with this disease.
Wonderful. And for all of this work that you're doing, you're really truly involved in many, many, promising and innovative work. So have you or are you in plans to integrate AI as you go forward with your mission? Because AI is doing some incredible, incredible things when it comes to patient.
Yeah, I obviously that's its own huge area. I think you know, I think the topic of AI is coming up at more and more scientific meetings where we go and people are sharing different ways to use AI. For me personally, so far it's been in digesting the large amounts of healthcare related information and scientific data and clinical data to make that more manageable and decipherable. I think in some aspects of medicine, AI is going to play a more hands-on role. For example, mammogram diagnostics or pathology diagnostics. I foresee these may be areas where AI might take the first pass, and then I think there's always going to be a clinical person that's going to sort of review and confirm results. But for cancer, I don't think we're quite there yet. But I think that's also a great topic to longitudinally see over time how it evolves, because I'm sure in our lifetime that's gonna bring great advances to medicine.
Yeah, and I was going to say, through your Breakthrough Cancer Initiative, I wonder what MIT Labs what they are going to say about the inclusion of AI into this whole project. So I've asked you a lot of questions, Dr. Jazaeri, as always, and I never finish my list because there's so much to learn from you. What have I not asked, like big and bold, that you would like to share with our audience today?
I don't know. I think we covered quite a bit. But certainly I love chatting with you. I love interacting with overcomers, and I look forward to future opportunities to update everybody on what's going on.
Okay, great. And then the last question that I always ask is, what message of overcoming would you like to share with everyone listening?
I tell all of my patients that without exaggeration, there is more hope today than ever before in the history of ovarian cancer. And so I see our job as keeping our patients going with currently available treatments. There's tons of research going on that are going to be tomorrow's treatments. That's not just a message. I can point to specific treatments that are available today, like Elahere and antibody drug conjugates, and even PARP inhibitors. These were not available 5 years ago. And so I want overcomers to definitely keep hope and know that there's lots going on. And even if a fraction of that proves to be helpful, that's still a much brighter future than ever before. So.
And perfect. And thank you so very much, Dr. Jazaeri, for all your wonderful insights. And you answer all my questions with so much details, and I just absolutely love it. So thank you so much for your time. And overcomers, let's end this episode on this high note of hope, as Dr. Jazaeri just mentioned. And we will be back with the next episode of Connect Over Coffee very soon. Until then, you keep inspiring and keep overcoming. Thank you and bye.